Good morning, everybody. I am Luisa Torsi, Professor of Chemistry at the University of Bari, Italy. Actually, right now, I'm also the president of the European Material Research Society. It is my great pleasure to chair this session at this very interesting conference. The title of the session is Material Science for Nanomedicine. And the first speaker is Professor Heinrich Hofmann, Director, Power Technology Laboratory, Institute of Materials at EPFL Lausanne. So.
Good morning. Thank you very much for the invitation. And now, so I will speak today about iron oxide nanoparticles. You see, the title is relatively short because I like to give you, let me say, a summary of impression on some conclusion what I have done from my results of our work during the last years. And then I will show you then also some new results, but much more, I think it's at the moment important to make some conclusion regarding iron oxide nanoparticles for medical application for sure. Yeah. So if you see the iron oxide nanoparticle, this is one of the few inorganic nanoparticles where we are using really the nano effect. That means if you go a very small particle around 10, 20 nanometers, then you change, as you know, the magnetic properties, so you get new properties, superparamagnetic properties, very important for imaging, for the colloidal behavior of the particle. If you go larger particle, then you go to a single domain particle, which has a very high magnetic saturation, and also, but they are permanent magnets. And then we have the multi-domain particle, which you know. And if you look now here, the application of this particle, so we have the imaging here. Yeah, the very small particle, superparameter are for imaging, MRI, contrast agent. Then we have for magnetofaction, this means more or less transport in vitro, yeah, not in vivo normally. Then we have also the very interesting application of magnetic hyperthermia. So if you change the size, you change the magnetic properties. At the same time, you change also the biological properties. So that we have here tumor penetration, accumulation. Then we have also fast endocytosis if they are a little bit larger. That means we have now to find the optimized, so they have to make also a compromise between the size, looking for the application means physical properties of the materials. At the same time, we have to look what happens in the body, which size is the right one for the application. If you like to go in tumor, or if you like to look for endocytosis, what else is not the same size. But we have to be also careful. Yeah, here in such a scale, we are speaking about the size of the core, and we speak the size of the particle itself. It's not the same with all the coatings around. So iron oxide nanoparticle is not new. There is a long history, yeah, and all this what you see here, the most are used for as contrast agent. And maybe you know that this particle were on the market in the clinic, but they disappeared from the market, and the reason was not safety reason, the reason was simply economic. Yeah, because there was only used, or mostly only used for the liver, yeah, imaging, and this is not the right market, not enough. There are some problems also with the kidney sometimes, but at the end, the market was not good enough for these particles, yeah, not large enough, so they disappeared. Yeah. We have at the moment, beside this ferroxytol, which is for not really a contrast agent. Yeah. Then we have also the other one now, you know, in Nijmegen, this SPL Medical is now producing again CLINAM type or Combidex type particles. So we have still this typical iron oxide contrast agent on the market. So the iron oxide nanoparticle is for sure a very complex system. Yeah, we have here the core, and we'll speak quickly about the core. Then we have the coating around, and with this we have then all these functional applications. So we have then, you see here other coatings and and functionalization, so that we have here biomolecules on the surface depending for the targeting, we have for signaling, and we have other functional groups on the surface. So at the end it's a very complex system, and this makes also is one of the reasons why we have still this huge barrier to bring this on the market, because you have to prove that this is really producible and can be produced in a good way. So here, speaking now of the core, one of the big problems was to get different sizes of this nanoparticle without organic solvent, without additives. Because we need particles which are more or less naked, then we can start to make a procedure for the coating and functionalization. So we have here a process developed to get particles coming up from 6 nanometers up to, let me say, 20 nanometers. Okay, they are not so nicely shaped as normally in organic solvent, but they are absolutely scalable. We can upscale it. The product process is GMP compatible. We have high flexibility in coating and functionalization. Very important. Then also here some results, you see, because here we have the size of the particle now on the core, because there are the magnetic properties. We have here for imaging, you can see here, this is Simmerem type particles. Here are the larger particles. We can have a much higher relaxivity. That means much higher contrast with such particle. If you go, for example, for hyperthermia, again, you can see that with larger particles we get higher specific loss power, so we can have higher heating, or we need less particle for the heating. Yeah? But still, we have to be careful with the numbers here. This is a typical area of the applied magnetic field and the frequency, which we can apply to humans. So it's very low, maybe 100 to 300 kilohertz in the area of, if you speak in Tesla, maybe 3 to 10 Tesla. These are areas here. Yeah? And if you go to rodents, you can have maybe this log scale. You can see here we can have a much higher, 100 times higher heating. But this is not what we can then apply to humans. So we have to really optimize the particle in these areas which we can apply to a human body. The rest here is not really of interest at the end. For scientific reason, okay, but not for application. So we have to look here, and then if you look on this here, we have SLP or SR values which are very low, which are in the area of 8-9 watt per gram. And this we have to optimize. So then we look now on clinical trials with coated particle, and now we come to the problem. There is not one clinical trial with functionalized nanoparticle. Yeah? All what you see here, all these trials are mostly with dextran modification coated particles. Yeah? For applications in the lymph node detections, head and neck cancer detection, cardiovascular disease, autoimmune disease, a little bit, yeah, cell tracking, stem cell tracking, yeah, delivery, not a lot, yeah, mostly gene delivery. Anemia is one of the most interesting area which is used since a long time, but in this case it's not really, in this sense, nano, yeah, because they are not using physical properties which are coming from the nanoparticle. Here is a very small iron, 2-3 nanometers iron oxide, which is there. We can also call it an iron cluster. And then you see here a little bit hyperthermia. So we have, these are all dextran-coated particles, yeah, and we have no functionalization. Functionalization is very important. Here we have here posters here from a project that is running together with different universities. One is from EPFL, the other one is from University of Geneva in this area. But you see also, if you look in the literature, we have here a lot of different coatings, for sure, because we are targeting different molecules and different cancer areas and so on. Interesting is to see, if you go in the literature, yeah, nanoparticle and coating functionalization is more than 500,000 papers. Yeah. If you look on SPION, the supermagnetic iron oxide nanoparticle and coating functionalization, we have only 280. Yeah. So still there's a lot of work to do to understand what happens with these particles. Yeah. Toxicity is similar. There's a lot of investigation regarding toxicity. we can say that generally that with small iron oxide nanoparticle we have maybe a little bit more reactive oxygen species and then genotoxicity of the reason of this. Yeah, we are the only small problem is that we have degradation of iron oxide particle. Yeah, and then maybe we have this femton reaction and maybe DNA damage, but this is not happen if the particles are in the liver normally. No? But anyway, all this tox stuff is strongly influenced by the type of coating and oxidation state of the iron oxide nanoparticle. Yesterday there was in the afternoon a very good session about toxicity of nanoparticle, and there was also stated that extra-coated iron oxide nanoparticles are not toxic. At least not for that aspect what they investigated, cytokines and others. Then we have, effect of actin skeleton and so on. But in vivo, there is no toxicity in vivo with dextran-coated nanoparticles, so we are relatively safe. But still, all this depends from the protein corona. I don't go in detail in this, but again, you see here, there is not a lot of literature regarding protein corona of iron oxide particle in vivo. It is only what is counting at the end, and because we have a lot in vitro, but what we are looking for, and this is, if you are interested for this, you have here in the afternoon a session about difference in vivo in vitro to go, I know, I'm finished in 1 minute, and so that we have now the outlook. So, Combidex is back on the market, so we have particles for imaging. Maybe further functionalization also. Then IOPs can produce in a simple aqueous method, showing high reactivity and high specific loss power. So they are highly biocompatible. So from this side, everything is okay now. But the problem is still we need increased specificity in targeting of cells. Which is shown on lab scale, is function, is possible, but still we have no clinical trials. So we need much more simplified coating and functionalization, which are at the end reproducible, and we can do it by correct scale-up production. So at the final is my acknowledgement, especially to Deborah Baumer and Mariana Mionik. Both has worked a lot in this area of synthesis of the particle and functionalization and protein corona of these biomes. And I'd like to thank you for your attention.
Thank you, Professor Hofmann. The paper is open for discussion. Any question, comment? No, so I think I'll start. Yeah, I was wondering, about the chemistry of surface chemistry and clearly electrochemistry of iron, and how it comes in when we talk about functionalization. I mean, I think this is extremely important. The results are very convincing. I was wondering how far we are with the development of, biofunctionalization method for this.
Yeah, we have a different method. So let me say, if you have go for direct organic coating. So functionalization means first we need a first layer, which makes the whole stuff biocompatible and colloidal stable, because iron oxide has an isoelectric point at 7, so they coagulate anyway, aggregate in the blood. So we have to stabilize this. Second is, and this must be, this we can do with phosphate groups, for example, which binds relatively good to the particle. On the other hand, then we need further functionalization. That has done a lot of work, you are right, to bind then the aptamers, the antibodies on the particle. There is also a lot of work is done. I think the problem there is reproducibility, and upscaling. This is the problem what we have. I think the chemistry is so far is fine. It's not a problem. Let me say, as a material scientist, I'm not a chemist, but for me, the chemistry is, I have the impression, is fine. The problem is really the reproducibility and to use this in higher amount and still stable. So we have a lot of problem to stabilize the nanoparticles colloidally in the blood and serum. And this is the main problem.
Thank you. Other questions, comment? Yes, please.
Could you state on toxicity, let's say, of the nanoparticles, the ROS production? Because it looks like that there are some indications that this is strongly cell dependent, that there are some cells which are producing, for example, transferrin, and so there's no ROS production, while others are unable to produce on time enough. Is it pathway dependent? So let's say, is there a difference if they are entering in the endocytotic lysosomal pathway or if you're entering in receptor-mediated uptake?
I think so. My impression, I have no final clear answer to this, but I say if you look on all the results what we have, if you have endosomal uptake, then it's not really a problem. Then mostly we have then, then we have not this ROS production. If the particles are in the liver, normally we have not a problem. But the main thing is we need Fa2O3 if possible, and not Fa3O4, because this is very important. And even we reduce the magnetic properties, but we gain on the biocompatibility way. And what we have also observed in vivo is with this ROS and maybe other immunotox is that in injection, in big animals, sheep, for example, between 24 hours and 72 hours, we have a light inflammation, a little bit inflammation, a little bit fever, but then this disappears. I think this is still, for a treatment, is acceptable. If it's acceptable for diagnosis, this we have to discuss. But still, we are on the good side.
Okay, very good. And thank you so much. In the interest of time, I think we shall move on.